you guys know, today's theme is the tools we use for science. So I'm going to tell you about 3 tools that I'm using in my research here at SDSU in Todd Anderson's Fish Ecology Lab. So I'm going to tell you what lasers, satellites, and ear bones tell us about fish populations. So the main big question of my research is to understand what factors affect fish populations. How are they changing through time and why? This is obviously really important for anyone who likes to eat fish. We want these fish populations around in the future. And so what we found through lots of research is that the larval stage is the key life stage for understanding how fish populations change through time. And this is because we have these adults that produce hundreds of thousands of these larvae, and about 99% of them or more are going to die. So if we can figure out who are these survivors, what are the characteristics of these survivors, or what were the environmental conditions when these guys were alive that made it, that can help us understand how these fish populations replenish through time. So what's going on during this bottleneck? So I'm studying rockfishes as a case study. So the rockfishes, which you can see here, here's an adult, here's a bunch of little babies. They are really good representative organisms for understanding all fish. They have a lot in common with a lot of the different fish that live on our coast. But also, rockfishes alone are really important to our ecosystems. So as babies, like these guys, they're really important food resource for fish like salmon. So if you guys like salmon or seabirds, a lot of animals will feed on baby rockfish. They're also, as adults, really important predators, so that's important for ecosystems. And we also like to eat them, so we have a lot of people who make a living by fishing rockfishes. So we're really interested in understanding their population and how to manage them. So now I'm going to tell you a little bit about this larval stage. Here we have an adult. She produces hundreds of thousands of these larvae, and they'll eventually migrate offshore to feed and grow. And the reason why this offshore environment is really a great place for a larval fish to live is because there's, in the springtime, these winds will start blowing that mix up the ocean, bringing nutrients to the surface and resulting in a bloom in productivity. So here you can see this bloom in productivity forms each spring of each year. So these fish will grow, they'll eat, and they'll eventually go through this process of metamorphosis where their body will actually change its shape, and then they're ready to go back to their nursery grounds, back to the kelp forest where they like to live. So now they have to figure out how to get back 50 kilometers back to shore. So they might be using some oceanographic currents to help deliver them back to shore in the process of settlement. So in my research, I'm really trying to figure out what's going on during this entire cycle. We also know that whatever happens during this larval change, whatever happens to their growth is really important. So we know that even small changes in how fast they're growing can have huge consequences for survival. So if they can grow just a little bit faster, they're way more likely to survive. So we really want to figure out what's going on with growth. So now I'm going to tell you about the first tool I'm using, which is a fish ear bone called an otolith. So here's a little baby fish. We can pull out these ear bones that sit right here in their skull, and here's what they look like. I can actually put them on microscope slides and sand them with really fine sandpaper, and that allows us to see all these rings. So this looks like a tree's growth rings, and that's exactly what it is. We can figure out the day each fish was born by a circular mark that's left in the ear bone. We can also count the number of days or the number of rings to figure out how old each fish was. And we can also figure out when it went through metamorphosis because they create this mark in the bone that signifies when it transitioned from that larval shape into this juvenile shape. So I'm going to be using these to measure all sort of characteristics about fish. And you can actually see these outside of the fish lab activity. So the second tool I'm using are satellites. So we have these satellites that are floating around in space, and they're taking pictures of the ocean. And scientists have figured out a way to convert these color images of the ocean to measure things like the temperature of the water and how much chlorophyll or primary productivity is in the water. So these satellites download all this data, and then I can figure out, is temperature or the temperature of the water that they're living in really important for their growth? Or is it the amount of food that's out there that's important for their growth? And I can actually tell the satellites the exact dimensions I want it to measure. So I know that larval fish live along the continental shelf, and I know that they're probably moving somewhere around 200 kilometers. So I can average those values in the exact area that I'm interested in and figure out if that predicts the growth of these fish. So, oh, I already told you that. So what did I find? Well, what I found is that the primary factor that influences growth of these fish is in fact the index we have for primary productivity. So it wasn't temperature that it's affecting their metabolism. It wasn't as much how fast they were growing right when they were born. The most important factor was food. So we know that these large-scale wind patterns that stimulate ocean productivity on a very large scale are affecting the growth and likely the survival of these fish. So I did this for a couple different species, and this is now also tells us that we can use these satellites that are taking pictures from far away, and maybe that can help us get a good idea of how fish populations are going to do each year without actually having to go out and catch them. So now I'm going to tell you about my third tool. I don't know what that slide is, how that got in there. So lasers. So how can we use lasers to find out about fish populations? So here we have, this is what actually a laser looks like. And the beam is right here. And I can put in these fish bones into the laser, and I can measure the chemistry that's recorded into these bones. So we know that if we sample, these are actual laser spots that I've sampled the fish bone, and this is the edge of the otolith, so that's the day I caught it. We can travel back in time and measure how the elements change in concentration through time. So this can tell us, are fish swimming through different types of currents or different types of seawater? And that helps us understand their movement or their migrations. So this is something that I've had to validate in my own research to figure out if this tool works. This has been a huge part of my research. So the way I've done this is collected all sorts of seawater over 4 years and tried to simulate natural conditions in the lab. So here we have, you can imagine, a migrating larval fish is going to experience really different kind of wind conditions that are mixing up the ocean. We're going to see all sorts of different temperature and all sorts of different salinity. And so I reared fish under these conditions, and then I tracked their seawater and their bone chemistry through time. So here's what I found. In the blue, you see that the water is starting out with a low concentration of this element barium, and then it increases. So we see a change in the type of water. And then we also see that the fish bone chemistry is actually matching that of the water that they're in. So this experiment is what validates that this tool can work for looking at fish movement. And the next step is I have several years where I've collected these fish in the field, and so now I can be able to measure their bone chemistry and figure out which currents they're using to get to shore. So that's to be determined. So what did we talk about today? Okay, so I told you that ear bones can tell us how fish are growing and how they might be responding to environmental conditions. We also talked about how satellites photograph the ocean and allow us to measure things like temperature and the productivity and the amount of larval food. And then I also told you how lasers can be used to measure the chemistry of fish bones so that we can understand how they might be associating with ocean currents for their movement. So with that, I will take any questions.
minerals from the area, and that's how you guys are able to figure out what it is from the laser readings?
So the bones is like a calcium carbonate. It's like a shell. Like when you touch a shell, that's calcium carbonate. That's their ear bone. And so it's growing each day, and it incorporates the elements in the seawater around it into its bone. And then the laser actually will like sample a tiny circle that's like 3 microns. So this is super small. And it will basically just explode that bone fragment into this air, and then the air, the bone gets ionized, so it breaks down into individual elements, and then there's another, it flows to a special machine that measures how many of each kind of element is in there. And so, and we do the same thing for the seawater, so it'll kind of like explode the seawater and it'll measure everything that's in it, and we can see how they match up. Yeah. So like for fish, when they're babies, they grow each day. And then eventually they start getting really, really big, and then we stop seeing the daily growth bands and they turn into annual growth bands. So when we collect an adult fish, we can pull out their larger otoliths and actually figure out how old they are in years. So that's how all fish research, when they measure age of fish, it's usually from their otoliths.
With the same rings, just like a tree, you can determine how much food and nourishment it gathered when it was growing by the size of the ring.
Basically, the distance between the rings tells us how much it grew.
So if it had plenty of food,
it'll be a thicker band. But I know the conditions because I know the day it was born, because I can figure, I know the day it was caught, and then I count the rings back. So I can say, okay, this fish was out there March 15th till July 3rd, and then I can go into my satellite data and average what was going on during that specific time. And so that's how we figured out all the pieces to the puzzle.